Bar has been raised by first the teaser, then Dr. Tiwari, and the previous topic. Now, after this clinical setting to the future and think about a very pertinent disease, tuberculosis. You're consultant in Department of Respiratory Medicine in Max Hospital, New Delhi. So I'll try and skip couple of things which I expect we all know about this. So the tuberculosis is by Mycobacterium tuberculosis. There are a lot of other species also, Africum bovis, which can be pathogenic to human beings. The burden of tuberculosis is the annual incidence is about 9.6 million globally, and India the incidence is about 2.2 million. And this is the current theory of mycobacterial infection, that we inhale this bacteria and then they are ingested by the macrophages, and which swallows the mycobacterium and invades and killing the bacteria. And the new theory suggests that the mycobacterium are inhaled and they enter into the body directly through the M cells, the microfold cells that line the airway tissue, and then they travel to lymph node and beyond, and then they get reactivated. A simple algorithm for the diagnosis of the tuberculosis. So you have this active tuberculosis which we deal almost every day, and half of it is diagnosed by history and clinical examination, then the radiology and the bacteriology. There are certain latent infection which are which we not bacteriological examination. Then we have these conventional diagnostic methods, the smear and the culture of the sputum. There are certain molecular methods which not only detects and identify the species of the tuberculosis, but also detect resistance in diagnosis, which in Indian setting are not methods. The expert in the MTB and rifampicin for MDR-TB is a newer PCR based diagnostic, and WHO recommended, currently used in this method in MDR-TB. It simultaneously detects the tuberculosis as well as the rifampicin drug resistance, which is of immense importance because you get the answer in accurate results in less than 2 hours. And it is pretty sensitive also, especially for rifampicin resistance. Now there are certain examination. It is difficult to procure the sample. There are false positive results. That means when the patient is recovering. Now what happens is that either you diagnose lesser number of patients, which means that the patient would continue to suffer and there would be increased mortality and morbidity in these patients, and the patient would continue to transmit this disease, or you diagnose overtly, giving the unnecessary treatment to the patient and leading to various other toxic effects of the medication, or you have a delayed diagnosis of the drug resistance. That means you are giving the drugs that are not effective against the disease. So therefore there is a huge gap between, considering the current limitation. The WHO has released a consensus diagnostic which are easy to run and can be taken care in the periphery also. They needs to be developed. So what are these? So there are a couple of tests which are still in clinical phase trials. Second line anti-TB drug assay. So these are the tests which have got various advantages, and skip these. Now this is phase. All those patients who fail or those who relapse or they are sputum positive after they have stopped the treatment come in category 2, in which there is an introduction of aminoglycoside streptomycin. The smear negative extrapulmonary tuberculosis or smear negative pulmonary tuberculosis which are not ill are classified under the latent. There we gave only HRZ for 2 months followed by 4HR. And then there is a chronic and MDR tuberculosis, for which the specially designated standardized or individualized regimes are given, which shortly I'll discuss in brief. Now, in this MDR tuberculosis, first I would like you to pay some attention. So you have the first line oral anti-TB drugs, so standard 4 drugs, which everybody knows are the aminoglycosides, streptomycin, kanamycin, amikacin, capreomycin, viomycin. Then you have the quinolones, the moxi, gati, levo, cipro, ofloxacin. And then you have the oral second line anti-TB drugs, which are not normally used, and they are ethionomide, prothionomide, cycloserine, terizadone, and PAS. Then you have certain other agents which have got a very unclear efficacy, but they are known to be anti-tubercular in certain sense. Then they are clofazimine, the drug used in leprosy, the good antibiotic linezolid, amoxicillin, clavulanic acid, imipenem, and high dose isoniazid, as well as clarithromycin, and these are the agents which have the unclear efficacy for pulmonary tuberculosis. Now what we do is that the treatment duration for this MDR tuberculosis is spanned up to 18 to 24 months with the effective drugs based on the sensitivity pattern. Now it's a mix and match. In the intensive phase, we take 6 drugs. So H and R are out because they are resistant. So we pick up E and Z from the group 1. And then we pick one injectable based on the sensitivity, obviously. And the streptomycin is usually avoided if the patient has already taken that. So we pick up one aminoglycoside from group 2, a fluoroquinolone from group 3, and 1 or 2 drugs from group 4 and 5. So usually it would be ethionomide with cycloserine, or ethionomide with say linezolid or clarithromycin, depending upon the severity of the illness. And these are given for 6 to 9 months. In between, you would continue to examine his sputum with not only with stain but also with AFB culture at periodic interval at a designated lab. And then after that, there will be a continuation phase for 18 months with 4 drugs. So again, 1 fluoroquinolone from group 3 and ethambutol, and 2 from group 4 and 5. They would continue for another 18 months. This is a 7 when the first sanatorium was introduced in Germany. Till now, we are still very far from TB elimination, and it remains a distant dream. The BCG vaccination was introduced in 1921, and it does prevent some disseminated disease in the childhood, but does not prevent or sufficient. The efficacy against the pulmonary forms of tuberculosis responsible for transmission of the disease is also variable. Now there are how to improve the BCG vaccination. So there are some subunit vaccines which can be used as boosters, and there is also focused on development of new, more effective live vaccines which are capable of replacing the current BCG. So these are all in clinical trial status right now. So shortly we may see these kind of subunit BCG vaccines in market. And here is one designated to boost the immunity primed by the BCG. Rifampicin in 1960, we have hardly had any anti-tubercular drug. The new shorter chief say there are 2 drugs in particular, the bedaquiline and delamanid, which I would discuss about this briefly. This bedaquiline is selectively targets the proton pump of ATP synthesis, and it is approved for MDR-TB. And similarly, the delamanid is nitroimidazole class of antibiotic, and it inhibits the synthesis of mycolic acids, and it is also approved for MDR-TB. It's trickling here in India, but in the pipeline. There are some other promising drugs, some that targets the cytochrome B subunit, some target the, some has the potent bactericidal activity against the XDR-TB, and then there are drugs which are active against both the drug-susceptible and drug-resistant tuberculosis by targeting the mycobacterial membrane protein. And the SEQ9 is in phase 2 clinical trial after assessing the safety and efficacy, which was completed. Now, there are some updates of the newer WHO guideline for MDR-TB, in which they have regrouped these drugs. So we have fluoroquinolones, we have second-line injectable drugs, we have other second-line drugs, and we have add-on agents. So now what do? We, because of this reclassification, the duration of the treatment is reduced from 18 to 24 months to 9 to 12 months. And so, during the intensive phase, a regime with at least 5 effective TB medicines are used, the pyrazinamide and 4 core second-line TB medicines, again fluoroquinolones, aminoglycoside, and 2 other from core second-line agents. And if the minimum effective TB medicines cannot be composed as above, the agent from group D2 and other agents from D3 may be added to the regime. Going ahead, India is the highest TB burden country, accounting for nearly 1/5 of the global incidence. And continuous research and ongoing tuberculosis across the globe to support WHO and TB strategy. We need a better understanding of the disease pathology and development of new diagnostic method, vaccines, drugs at the various stages of development. And we need them now. And here is one promising thing from the House of Cadila, which I would brief. The hidden facts, there are certain hidden facts about the rifampicin, that it induces the cytochrome P450, and it also leads to it lowers the bioavailability in rifampicin and leads to subtherapeutic rifampicin level and slow response to the treatment. And here you can see, in fact, the bioavailability of rifampicin reduces to 68% after 3 weeks. The stability of rifampicin can be enhanced by, so isoniazid releases itself with rifampicin, then it reduces the bioavailability of the rifampicin. And therefore, if the isoniazid can be stabilized and can be made to release itself in duodenum, then it increases the bioavailability by 90%. And then there is this magic potion, that is the piperin, the bioenhancer. So highly purified and standardized extract obtained from Piper nigrum and Piper longum. It enhances the bioavailability of the number of drugs, including the rifampicin. And how it does this? Because the rifampicin induces P450, and therefore this piperin inhibits the cytochrome P450, thereby decreasing the metabolism of many drugs, including rifampicin. So piperin inhibits what the rifampicin potentiates. So the integration and degradation of rifampicin can be reduced, and stability can be enhanced by enteric coated of isoniazid, and Rizorin is the only brand with the patented drug delivery process. Which suggests that in Rizorin the rifampicin 450, and there is a clinical study to support this. Thank you very much.